one question and then go. Hello, students, and welcome to the final session of NPTEL DA sessions. This is the course Basics in Biology NOC 25 BT02, and we'll be dealing with lecture week 12. This is the final lecture week with 4 lectures. So all these questions will be including the topics taught in all those 4 lectures. The course instructor for this course is Professor Vishal Trivedi. He's from IIT Guwahati, and my name is Ashutosh Behra. I'm a PhD student from IIT Kanpur. So these are the basic rules that we'll follow through this presentation. I would request all the participants to please switch off your mic and video chat and interact with me through the comment box. So what we'll be dealing with is 15 questions, multiple choice questions from these topics, and I'll post them one by one, give you some time, and we will discuss the correct answers. And you, at the end of the minute, you can post your correct answers or whatever answer you think uh, is the legitimate one in the comment box, and I'll be checking through it regularly. So let's keep this session interactive, and this is a recorded video, recorded lecture. So all those who will be watching this later, I would also request to participate in this interaction. Okay, with that much of an introduction, let me present my slides. Yeah, I think you can if you can see my slides, just let me know in the chat box with a yes or no or a like. Can you see my slides, and is my voice audible to you? Okay, great. So hi everyone, and I welcome you again to week 12 for Basics of Biology NPTEL. This is the final TA class, TA live sessions for this module. And after this, there was a request from NPTEL authorities for an extra class, which will be held on Tuesday, the same time, 3 to 5 p.m. So if you have any doubts, please ask your friends and do join the live class. Okay, and my name is Ashutosh Behra again, introducing myself again. I'm a PhD scholar at IIT Kanpur. So starting off with the first question of this session. Question number 1. The body of the earthworm is divided into segments of serial repetition with few organs called as dash. Read the question again. The body of the earthworm is divided into segments of serial repetition with few organs called as dash. Options: option A, metamerism; option B, isomerism; option C, symmetry; and option D, pseudo-metamerism. So these are the 4 options. If you remember the first week lectures, we read about the different kingdoms, different classes of animal kingdom, and earthworm is a segmented animal. What is this phenomenon, serial of repetition serial organs of the earthworm known as? Let's take a minute of time to collect some answers. Please post your answers in the chat box, and I'll reveal what is the correct answer at the end of the minute. time's up. That's enough time for this question. The body of the earthworm is divided into segments of serial repetition with few organs called as, and the correct answer to this question is option A, metamerism. So metamerism is known as repetitive segmentation of the earthworm body. Let's learn about metamerism in earthworms. As you can see, the definition of metamerism in earthworms refers to body's organization into series of repeating similar segments called metameres, also known as somites. So we see a similar kind of organization in the embryonic stages of mammals as well, where we have somites giving rise to the arising from the mesoderm and giving rise to mesodermal tissues. These segments are separated by internal partitions called septa, and each metamere contains repeated structures like muscle groups, nerve ganglia, and excretory organs. So here is a diagram on my right showing the earthworm segmentation, and the internal segments are separated by partitions called septa. Septa means partitions, and this is the prostromium peristomium, which is the mouth of the animal, and the tail of the animal here. There are several septa which is dividing the body of the earthworm into segments. This segmentation allows for greater flexibility and independent movement of each segment, aiding in locomotion and burrowing. So this segmentation is important for the animal's lifestyle and habitat, which helps the animal for locomotion and for burrowing into soil, for which earthworm is known for. Now, what are the other options? The other options were isomerism, symmetry, and pseudometamerism. Isomerism refers to the phenomenon where compounds have the same molecular formula but different structures. In biology, it can refer to symmetry or balance in body parts, but it's not relevant to segmentation of the earthworms. So if you have left and right symmetry, which is almost isomerism is used for chemistry related term. What is symmetry? This refers to balanced arrangement of body parts such as bilateral or radial symmetry. Earthworms do have bilateral symmetry, but this term doesn't describe segmented structure of the repetition of organs. So earthworms do have the bilateral symmetry. So as we move ahead in the or higher in the animal kingdom in the classes, we see a longitudinal bilateral symmetry, and in lower organisms they have radial symmetry. As you can see on my right is a Drosophila fruit fly with a bilateral symmetry, flowers of the angiosperms with a radial symmetry, and a bacteria with a spherical symmetry. And what is pseudometamerism? This term refers to a false or superficial segmentation that doesn't involve true repetition of internal organs. So what happens in a metamerism is each segment is repeated truly to its nature, and each segment has a repetition of all the organs. May it be nerves, muscles, or blood vessels, each segment has a particular arrangement, and such an arrangement is repeated over the body, causing a true segmentation. And what is pseudometamerism is not a true segmentation but a false segmentation, does not involve repetition of internal organs. However, this is also seen in some animals like tapeworms, which belong to Aschelminthus and Platyhelminthes. Earthworms, however, have true segmentation with repeated organ system, so this does not apply to earthworms. So the correct answer was metamerism, which is segmentation of body parts of the earthworm and forming repeated structures. Each segment has a particular set of structures which is repeated in every segment of the earthworm body. Is it clear to the students? Just let me know with a yes or no or a like in the chat box, and we can move ahead with the next question. Sorry students, I think there was a network issue and my screen got locked out, maybe. So I'll try to present again, share my screen. Sorry. Just give me a moment. I'm trying to reshare my screen. Yeah. I hope you could see the first question that we were trying to solve. Metamerism in earthworms. We were just about here. Yeah, I think this was the point where I logged out due to poor network issues. So as I was talking about, all the other options were isomerism, symmetry and pseudo-metamerism. Pseudo-metamerism is a false superficial segmentation which is seen in tape forms, but earthworms have true segmentation with repeated organ system. Segmentation means each segment has a particular set of tissues, muscles, nerves and organs which is repeated throughout the body of the earthworm in each segment. Symmetry refers to the balanced arrangement of body parts such as bilateral or radial symmetry. We humans are bilaterally symmetrical. So these options do not click to the first question, and the correct answer is metamerism, which is true segmentation of earthworm body. Metamerism also helps earthworm in its living habitat for burrowing and movement. I hope it is clear to the students. Let me know in the chat box with a yes or no, or a like would also suffice. Okay, we will move on with the next question. Question number 2. The type of association seen between the humans and bacteria living in the intestines. This is the question number 2. The type of association seen between the humans and bacteria living in the intestines is option A, parasitism, option B, commensalism, option C, mutualism, and option D, competition. The humans and gut bacteria, what kind of association can be between them? Is it parasitism, commensalism, mutualism, or competition? Let's take a minute to crack this question, and I will reveal the correct answer at the end of the minute. Please post your answers in the chat box. I'll be following it routinely. Okay, I think that's enough time for this question. The type of association seen between the humans and bacteria living in the intestines is. The correct answer to this question is option C, mutualism. This is the correct answer. So when I say bacteria, these are nothing but the gut bacteria that is living in the large and small intestine, mostly large intestine in the humans. And it is a mutualism kind of an association. Now, what is mutualism? Mutualism is a type of symbiosis where two or more species benefit from each other. So, this is an interaction in which the human benefits as well as the gut microbiota. The bacteria benefits and its interacting partner, the human, also benefits. So, it's a type of symbiosis in which both the partners, or more than all, whatever partners are participating, are all benefiting from each other from this interaction. And the best example here is humans and gut bacteria. Another example may be bees and flowers. Bees pollinate the flowers and bees get nectar in reward. The pollen is also serving as a reward. Clownfish and anemone. Anemone provides protection to the clownfish, and anemone provides protection and shelter to the clownfish. And the anemone cannot sting the clownfish, which is resistant. And clownfish provides nutrients and waste for the anemone. Ants and aphids, lichen, which is a symbiosis between fungi and algae, mutualism, woolly bat and pitcher plant, all these are well known examples of mutualism kind of association. Now, what can be other kinds of association? Here is a table describing the interaction, the type of symbiosis, and a good example from these. Mutualism, species A and B both benefit. Species A is happy, B is happy. Prime example is a sea anemone and a clownfish. Commensalism, species A benefits but species B is unaffected. In the interaction, one of the species is benefiting from the interaction but the other is unaffected. Like a barnacle sticking onto the whale. The whale does not have any benefit of a barnacle, but the barnacle has benefits. So barnacle is species A and whale is species B. Parasitism, on the other hand, where one species benefits and the other is harmed. Like a tick on the dog's skin or fur. Tick sucks the blood from the dog. Tick is benefited but dog is harmed. So these are the other kinds of interaction that we saw in the options as well as symbiosis. But the correct answer was mutualism, which is between the human intestine and the gut microbiota. I hope it's clear to the students. If yes, just let me know in the chat box and we can move to the next question. Okay, great. Moving on to the next question. Question number 3. The absence of nucleolus. The absence of nucleolus is seen in dash. So which of these species would you expect to see no nucleolus? Mind the difference between nucleus and a nucleolus. And the options are option A, fungi, option B, archaea, option C, protista, option D, animalia. Which of these species, which of these kingdoms does not have a nucleolus? Let's take a minute and collect some answers in the chat box and I will reveal the correct answer. Okay, I'm sorry for that interruption again. I'm sorry for that brief interruption again in the flow of the presentation. I don't know why, but I'm having some discrepancy in the network connection. But we will redo this question. Question number 3. The absence of nucleolus is seen in dashed. Option A, fungi. Option B, archaea. Option C, protista. Option D, animalia. Which of these kingdoms, animals in which of these kingdoms or organisms in which of these kingdoms do not have a nucleolus? Post your answers in the chat box. Let me know of it. Okay, it's enough time for this question. Time to reveal its correct answer. The absence of nucleolus is seen in. Correct answer to this question is option B, archaea. Archaea are prokaryotes and they do not have nucleolus. The absence of nucleolus in archaea is a key feature that distinguishes them from eukaryotic cells. As I said earlier, archaea are prokaryotic, and the basic difference between a prokaryote and a eukaryote, one of the basic features is the absence of nucleus in the prokaryotes, while eukaryotes have well defined nucleus. Now, what is nucleolus and what is its function in eukaryotes? The nucleolus is a dense region within the nucleus of the eukaryotic cells where ribosomal RNA is synthesized and ribosome subunits are assembled. So, nucleolus is a region for rRNA synthesis, and ribosome, which are the translating machinery, gets assembled in the nucleolus. Now, how does an archaeal cell structure look like? Archaea are prokaryotic organisms. They do not have a nucleus or other membrane bound organelles. So, this is a characteristic feature of prokaryote. They do not have any other membrane bound organelle as found in the eukaryotes, like Golgi apparatus, endoplasmic reticulum, vacuoles, lysosomes, nothing. Their genetic material is located in a nucleoid region not enclosed by a membrane. So they do not have a they do not have a membrane nucleus as such, but the region where the genetic material of a prokaryote is located is called a nucleoid. Despite lacking a nucleolus, archaea still synthesize rRNA and assemble ribosomes, but this occurs in the cytoplasm or associated with the nucleoid. So there is no designated nucleolus as such in the prokaryotic archaea. Now, what are the ribosomes in archaea? Archaea have 70S ribosomes like bacteria, but their ribosomal proteins and RNA polymerases are more similar to those of eukaryotes, a unique feature that makes them molecularly distinct. That is why they are molecularly distinct from bacteria and have formed a separate class of organisms called archaea. Archaea lack a nucleolus because they are prokaryotic. However, they can still carry out the functions associated with the nucleus, such as rRNA synthesis and ribosome assembly, just in a different, simpler way without compartmentalization. So they do not need compartment formation, they can still do the functions what the nucleolus of the eukaryotes does. So here is a comparison between all the organelles in a eukaryote and a prokaryote. There are membrane enclosed nucleus and a dense region called the nucleolus. There is a mitochondria, there are ribosomes in the eukaryote. But in a prokaryote, there is no such nucleus, but a region where the genetic material is packaged called the nucleoid. There is an outer covering around the cell membrane, which can be capsule in some prokaryotes, ribosomes floating in the cytoplasm, cell membrane, presence of cell membrane, and prokaryotes may also have cell wall.
which is only present in some eukaryotes like plant. Prokaryotes also have flagellum for movement. So these are basic differences between a eukaryote and a prokaryote, but nucleolus is not present in the prokaryotes. But all the functions can be carried out by archaea for RNA synthesis, for ribosome assembly. Thus, archaea is the correct answer, which does not have a nucleolus. I hope it is clear to the students. If yes, then let's move on to the next question. Question number 4. The force with which coacervates are held in the formation of colloidal aggregate is dashed. So this is pertaining to the origin of life theory, where we have coacervates. The force with which coacervates are held in the formation of colloidal aggregate is dash. Which is the force that plays a role in coacervate formation? Option A, ionic force. Option B, covalent force. Option C, van der Waals force. Option D, hydrophobic force. Which of these forces are necessary for coacervate to be held in the formation of colloidal aggregate? Basically, aggregation. Let us take a minute and find the correct answer for this question. I will reveal what is the correct answer in a minute or so. Okay, that's enough time for this question. Time to reveal what is the correct answer in this question. The force with which coacervates are held in the formation of a colloidal aggregate is, and the correct answer to this question is option D, hydrophobic force. Hydrophobic force is the essential force in formation of aggregates. Now, what is hydrophobic force and where does it arise from? Coacervates are formed through phase separation of colloidal substances like proteins, nucleic acids, or polysaccharides. The aggregation into coacervates is primarily driven by hydrophobic interactions, which occur when non-polar molecules cluster together to minimize their exposure to water. So, this is the origin of the hydrophobic force. Non-polar molecules, or molecules which cannot dissolve in water, like oil, like long chain hydrocarbons. If you have two such molecules which averse water or have phobicity towards water, they will try to minimize their interaction with water by interacting with themselves. So a non-polar molecule will readily interact with another non-polar molecule, making it a hydrophobic force so as to minimize their interaction with their water. These hydrophobic forces play a key role in the self-assembly of coacervates into colloidal aggregates, making them important in theories about the origin of life. So this is what I was mentioning about. In the origin of life theory, we speculate that these components of the cell, which are DNA, protein, nucleic acid, DNA, protein, and carbohydrates, have all aggregated from the colloidal solution as coacervates due to this hydrophobic force. So when there is no interaction and there are two such non-polar molecule, delta H is negative, delta S is negative, and delta G is positive. So this is an unfavorable reaction, not unstable reaction. Delta G being positive is an unstable reaction, whereas when there is hydrophobic interaction, there is delta H is positive, entropy is positive, and the free energy is negative. So free energy being negative is a stable state or a stable state of the reaction. So hydrophobic interaction or the hydrophobic force minimizes the exposure of non-polar molecules to water, sticking them together. And this is how we speculate in the theory of origin of life that coacervates have been formed when they are held by hydrophobic forces in the colloidal solution, coacervates of nucleic acids, proteins, lipids, and polysaccharides. So what happened in prebiotic earth? Some call it primordial soup. There were all these kinds of molecules, the nucleic acids, the NTPs, then lipids, protein molecules, some ions, phosphate, all together in the water, in the medium. Then came the coacervate droplets, which aggregated together. Fatty acids, nucleic acid, amino acid aggregated together. Fatty acids formed another phase. Inorganic salts and organic chemicals formed another phase. So they phase separated as coacervate droplets in the presence of the hydrophobic force, and then made an interacting coacervates interaction between the nucleic acid amino acid bubble coacervate with the fatty acid coacervate and the inorganic salt and organic chemical coacervate. Molecular exchange within the environment also happened between these bubbles. This led to what we now know as a cell or the origin of life. So the hydrophobic force has an important role in the theories of origin of life and understanding them. I hope it's clear to the students. If yes, then we will move forward and do so. Moving on to the fifth question. Who considered evolution as the hierarchical linking of series of forms in scala natura? Who considered evolution as the hierarchical linking of series of forms in scala natura? Option A, Carolus Linnaeus, Carolus Linnaeus. Option B, Charles Darwin. Option C, Robert Whittaker. Option D, Aristotle. Who of these scientists or philosophers thought that evolution is a hierarchical linking of series of life forms in scala nature? If you remember this well and good, or else you will know what is the correct answer in a minute. Please let me know what you think is the correct answer to this question. Post your answers in the chat box and I will reveal the correct answer in a minute or so. Okay, it's enough time for this question. Time to reveal its correct answer. Who considered evolution as a hierarchical linking of series of forms in scalar nature? The correct answer to this question is option D, Aristotle. Aristotle was a philosopher who penned down Scala Natura and who considered evolution as a hierarchical tree of all the life forms present on the earth. Let's take an example to understand it better, like the evolution of the brain. According to Scala Natura, the brain of a fish is the least, if you consider all these animals, fish, amphibian, reptile, bird, mammal, or human, the fish has the least evolved brain, and as we move up in the ladder, we have the higher brain because of the brain size. Then the amphibian comes up, the reptile, bird, mammal, human. But what is now known is that this is a modern approach, that there are branches to this tree. Fish clusters somewhere here, reptile and bird are more related, so they cluster here. Amphibians are here, and humans and mammals are more related, so they are connected here. Aristotle's Scala Natura, Latin for ladder of nature. So this is kind of a ladder of nature or great chain of being, is a philosophical concept that proposes a hierarchical arrangement of all things in nature, from inanimate objects to humans, based on their perceived complexity and perfection. So this Scala Natura, Aristotle wrote it to rank all these organisms or objects based on their perceived complexity and perfection. How complex is a structure is, how it is perceived to be complex, that was the key factor in ranking organisms or evolution in Scala Natura. What were the key features of Scala Natura that versus the modern theories? Linear versus branching. The Scala Natura views a natural world as a linear progression from lower to higher, whereas evolution depicts a branching tree life with numerous lineages. So evolution says the Scala Natura says that we have the tree of life, a linear progression from the lower to higher forms, whereas evolution depicts a branching of life with numerous lineages. Yes, sorry. Evolution will tell us that some of the species have branched and are closely related than those that are not branched or included in the same branch, right? No evolutionary history. The Scala Natura lacks an understanding of historical relationships and common ancestry, a central tenet of evolutionary theory. So it doesn't takes into consideration the historical relationships and common ancestry. Which one? The Scala Natura. No gradation of perfection. The Scala Natura suggests a progression towards perfection, with humans at the top. Evolutionary theory, on the other hand, emphasizes that evolution does not imply a hierarchy of perfection, as explained by the Kimela Center for Animal Advocacy. Anthropocentric bias. The Scala Natura often places humans at the apex of the chain, reflecting an anthropocentric perspective that is not supported by evolutionary biology. So humans cannot always be at the top right now. But maybe at that time, like some 200 years, some 200 years back, the Scala, not 200, more than that, the Scala Natura often places humans at the apex of the chain. Superiority for humans. See, here on my right is a pedigree of man, with man on the top, and this is a picture from Scala Natura. There are many organisms down the tree, and as we go up the tree, we see higher evolved organisms. So Aristotle was the one who wrote this Scala Natura. Aristotle is the correct answer to this question. I hope it is clear to the students. Let me know in the chat box with a yes or no, and we can move on to the next question. Okay, coming to question number 6. Euglena reproduce by the process of dashed. Euglena mainly reproduces by the process of dash. Option A, fragmentation. Option B, binary fission. Option C, budding. Option D, regeneration. Euglena reproduce by the process of fragmentation or binary fission, budding or regeneration. Let's take a minute to find out what is the correct answer to this question is. Please write your correct answers, write your answers in the chat box. Okay, that's enough time for this question. Euglena reproduces by the process of, and the correct answer to this question is Option B, binary fission. Euglena reproduces by binary fission. Let's see the binary fission in Euglena. So this is a mode of asexual reproduction in Euglena. Here on my left is a mature Euglena. First, the nucleus divides inside the Euglena. The flagellum also divides with its basal body. Then the cytoplasm starts dividing the nucleus into two. Then there is a fission point. Young Euglena forms, and there are two daughter Euglena. So binary fission, one parent dividing into two, exhausting the previous parent. So two daughter cells, one parent into two daughter cells. This is the form of asexual reproduction, binary fission in Euglena. Euglena reproduces asexually through longitudinal binary fission, where the parent cell divides along its length into two identical daughter cells. This process involves division of the nucleus, kinetosomes, and chromatophores, with one daughter Euglena retaining the parental flagella. Yes, I hope you could understand. Here is a more detailed breakdown. Initiation: a longitudinal groove develops in the middle of the anterior end of the Euglena cell. Then there is groove expansion. This groove gradually extends towards the posterior end until the two daughter cells are separated. Nuclear division. The nucleus divides by mitosis into two daughter nuclei. Organelle division. The kinetosomes and chromatophores also divide, and the flagella is also distributed. One daughter Euglena retains the parental flagella, while the other develops a new flagella from the newly formed basal granules. And the daughter cell formation. This process results in two genetically identical daughter Euglena cells resembling mirror images of each other. I hope that was clear to the students. Okay, great. Let's look at the seventh question of this session. Question number 7. The membrane strengthening molecule in prokaryotes is dashed. What is the membrane strengthening molecule in the prokaryotes? Option A, hopanoids. Option B, cholesterol. Option C, sterols. Option D, isoprene. Which of these is the membrane strengthening molecule in the prokaryote? Let's take a minute's time, minute or two, and think about it. Let's come up with answers. Post your answers in the chat box. I'll be following it up closely, and I'll reveal the correct answer in a minute or so. Okay, I think it's time for me to reveal the correct answer. The membrane strengthening molecules in prokaryote is, and the correct answer to this question is option A, hopanoids. Hopanoids are the membrane strengthening molecules found in the prokaryotic cell membrane. Now, what are hopanoids? Hopanoids are pentacyclic triterpenoids. Pentacyclic triterpenoids, a type of lipid molecule. So, these are a class of lipid molecule. They are structurally similar to sterols like cholesterol found in eukaryotic cell membrane. So, on my right is a comparison between a characteristic classic cholesterol and a hopanoid, which is a bacteriohopan E-tritol, E-tetrol, 5-membered ring, and a triterpenoid, pentacyclic triterpenoid. Now, these hopanoids are embedded in the bacterial membrane like this, plasma membrane of the bacteria, like this. What is the function of the hopanoids in prokaryotes? Membrane stability. Hopanoids help stabilize bacterial membranes, especially under stressful conditions like temperature and pH changes. They reduce membrane fluidity and permeability, providing rigidity much like cholesterol does in eukaryotic membranes. So, whatever the role cholesterol plays in eukaryotic membrane is a similar kind of role played by hopanoids in the bacterial membrane. Helps stabilize the bacterial membrane under stressful conditions like high temperature or pH, reduces membrane fluidity, permeability, and it is commonly found in cyanobacteria, actinobacteria, and some methanotrophs. Now, hopanoids are unique to prokaryotic cell membranes, only found in prokaryotes, especially in bacteria. They function similarly to cholesterol in eukaryotic cells, helping to maintain membrane fluidity and integrity. Cholesterol and sterols are typically found in eukaryotic membranes, not in the prokaryotes. And isoprene is a building block for certain lipids, especially in archaea, but is not specifically responsible for membrane strengthening in most prokaryotes. So, cholesterol and sterols are in eukaryotes, isoprene is in archaea. So, the correct answer is hopanoids, which is present in bacteria, namely cyanobacteria, actinobacteria, and some methanotrophs. So here is a diagram showing a cholesterol molecule and a glycolipid molecule in the animal or eukaryotic cell membrane. The similar function is provided by the hopanoid molecule in the cell membrane of the bacteria. Is it clear to the students? Just let me know with a yes or no in the chat box. Yes, very good. Here is the 8th question for you. The phase at which spindle pole position checkpoint arrests the cell cycle is dashed. I'll read out the question again. The phase at which spindle pole position checkpoint arrests the cell cycle is dashed. Option A, S phase. Option B, G1 phase. Option C, G2 phase. Or option D, M phase. Which of these phases in the cell cycle will be the spindle pole position checkpoint arrest? Let us take our time and find the right answer to this question. Please post your answers in the comment box. Okay, it's time for me to reveal what is the correct answer to this question. The phase at which the spindle pole position checkpoint arrests the cell cycle is, and the correct answer to this question is option D, M phase. It's the M phase of the cell cycle. Now, what is the cell cycle? The cell cycle is a series of events that a cell goes through as it grows and divides. It's divided into two main phases, interphase and the mitotic phase. Interphase includes G1, S, and G2 phases where the cell grows, replicates its DNA, and prepares for division, respectively. The mitotic phase or the M phase involves mitosis, which is the nuclear division, and cytokinesis, resulting in two daughter cells. So, this is the whole cell cycle in a nutshell. Series of events that a cell goes through as it grows and divides, and is divided into two main phases, interphase and the mitotic phase. Interphase is G1, S, and G2. Mitosis phase is mitosis and cytokinesis. G1 is the phase where cell growth happens. S phase is the phase for DNA replication. G2 is the another phase for cell growth. And at the mitosis, which is the end of the cell cycle in the M phase, there is the formation of two daughter cells. So this is the whole cell cycle. Now, what are cell cycle checkpoints and where does the checkpoint mentioned in the question come up? Cell cycle checkpoint, specifically the spindle checkpoint, also known as spindle assembly checkpoint here in the M phase or metaphase checkpoint, are crucial for ensuring accurate chromosome segregation during cell division. So these checkpoints check for the proper alignment and of the chromosomes at the metaphase plate. If the spindle checkpoint detects issues with the spindle assembly or chromosome attachment, it arrests the cell cycle, preventing the cell from entering anaphase until the problem is resolved. So if it detects any kind of changes or any kind of discrepancy in the spindle arrangement, attachment of the spindle fibers to the kinetochore, or alignment of the chromosomes on the metaphase plate, or anything as such, it will stop the cell cycle progression at this checkpoint until the problem is resolved. The cell cannot move into the anaphase. This arrest is necessary to prevent aneuploidy, unequal distribution of chromosomes, and genomic instability. So this is an important step to prevent aneuploidy. Now, not only spindle assembly checkpoint, there are many such checkpoints present in G1, S, G2 and M, all phases of cell cycle. G1 to S transition is a DNA damage checkpoint, where in G1 phase there is cell growth. Then inside the S phase is another DNA damage checkpoint where DNA replication is undergoing in the S phase. There are two checkpoints in the G2 phases, DNA damage checkpoint and the DNA replication checkpoint, and the G2M transition, which is antiphase checkpoint. So all these different checkpoints are present in the cell cycle throughout, but the most important is the spindle assembly checkpoint that can arrest the cell cycle and prevent it from going into anaphase if there is misalignment or arrangement of the chromosomes, attachment of the chromosomes to the spindle fiber, alignment on the metaphase plate, or any problem in the metaphase. I hope it is clear to the students. If yes, then let's move on to the next question. Question number 9. The first ever genetic recombination mechanism in bacteria was done by dashed. So this is an experimental based question performed by some scientists. The first ever genetic recombination mechanism in bacteria was done by dashed. Option A, J. Lederberg and E. L. Tatum. Option B, Avery, McLeod and McCarty. Option C, Frederick Griffith. And option D, Hershey and Chase. Come on. You should know the historic experiments, classic experiments which led to the discovery of the information and facts that we know about in our textbooks today. Which option is the correct answer? Who performed the first ever genetic recombination in bacteria? Who showed experiments? Who performed experiments to show that genetic recombination can happen in bacteria? Let us take a minute or so and arrive at correct answers. Please post your answers in the chat box and I will take a look at it. Okay, I think it's time for me to reveal the correct answer to this question. The first ever genetic recombination mechanism in bacteria was done by J. Lederberg and E. L. Tatum. These guys these guys won the Nobel Prize for this experiment, and this is none other than the famous conjugation system in bacteria. So these are the two guys. On my left is Lederberg and my right is Tatum. Joshua Lederberg and Edward L. Tatum conducted groundbreaking experiments in 1946 demonstrating genetic recombination in bacteria using E. coli bacteria. They showed that bacteria could exchange genetic material through a process called conjugation, where two bacterial cells form a physical bridge and transfer DNA. This was a pivotal discovery in bacterial genetics and earned them the Nobel Prize in Physiology or Medicine in 1958, shared with George Beadle. So they won the Nobel Prize for this experiment to explain bacterial conjugation, where there is a physical bridge formation and transfer of DNA from one bacteria to the other. So here is the whole process of a conjugation happening in bacteria. It is generally seen that bacteria can form, in the conjugation there is a donor cell which donates the DNA and there is a receiver cell which receives the DNA. The donor cell generally has F plasmid, so these kind of cells are called F plus cells, and the receiver cell is a F negative cell. Donor cell attaches to a recipient cell with its pilus. The F plasmid helps in the pilus formation. The pilus draws the cell together. The cells contact one another. One strand of the plasmid DNA transferred to the recipient. The recipient synthesizes a complementary strand to become an F plus cell. The donor synthesizes a complementary strand, restoring its complete plasmid. So once the plasmid, which is an extra chromosomal DNA material, replicates on its own to form a double-stranded plasmid, another copy of the plasmid, one of these strands can go into the F minus cell through the pilus DNA bridge made by the F plus cell, and then replicate inside independently in the recipient cell to make it an F plus cell again. So this is the whole process of conjugation, which was discovered by the work of Lederberg and Tatum, which won them the Nobel Prize, which also showed that genetic recombination for the first time in bacteria through the process of conjugation. I hope it is clear to the students. If yes, then let's move on to the next question. Question number 10. The most abundant neutral lipid stored in the adipocytes is dashed. This can be an easy question. The most abundant neutral lipid stored in the adipocytes is dash. Option A, triacylglycerol. Option B, diacylglycerol. Option C, monoacylglycerol, or D, cerebroside. All these are kinds of lipids. Which is the most abundant neutral lipid stored in the adipocytes or the fat cells in our body? Is it triacylglycerol? Is it diacylglycerol? Monoacylglycerol or cerebroside? Come on, quickly tell me in the chat box. Let's wait for a minute or a couple of minutes, and then I will reveal what is the correct answer to this question. Okay, that's enough time for this question. The most abundant neutral lipid stored in the adipocytes is option A, triacylglycerol. Triacylglycerol is stored in our fat cells or adipocytes in our body. Triacylglycerol or TAG, also known as triglyceride, is indeed the most abundant neutral lipid in adipocytes, which are fat cells. These cells store energy primarily in the form of TAG within lipid droplets, which are the main components of the adipose tissue. So on my right is a lipid droplet here with all these components of the lipid droplet. These contains phospholipid layered lipid droplet associated proteins in the phospholipid, and we have cholesterol, liquid triacylglycerol and solid triacylglycerol, which is there inside the liquid droplet. Now what are neutral lipids? Neutral lipids are those that are not charged or uncharged and include triacylglycerols, cholesterol esters and wax esters. Adipocytes and adipose tissue. Adipocytes are fat cells are specialized cells that store energy in the form of fat, primarily TAGs. Adipose tissue is a type of connective tissue made up of adipocytes. It's the fat tissue of our body. This is the structure of a triacylglycerol molecule. As the name suggests, the glycerol backbone with the 3 carbon triacyl is acylated with the long chain fatty acids. It can be saturated fatty acids, mono unsaturated fatty acid with a double bond here in the carbon chain, and polyunsaturated fatty acid with 3 fatty acid chains, right? So this is a general structure of a triacylglycerol, which is stored in the adipocytes as lipid droplets. I hope it is clear to the students. Great. Let's look at the 11th question of this session. The reducing agent that reduces the disulfide bridges in a protein into thiol groups is dashed. The reducing agent that reduces disulfide bridges in a protein into thiol groups is dashed. Option A, beta mercaptoethanol. Option B, guanidine detergent. Option C, urea. Option D, alkalis. Let's take a minute to find out this answer, and I'll reveal the answer in a minute. Okay, it's time for me to reveal the correct answer to this question. The reducing agent that reduces disulfide bridges in a protein into thiol groups is beta mercaptoethanol, option A. Option A is the correct answer, beta mercaptoethanol. Now, what is beta mercaptoethanol? This is a reducing agent that breaks the disulfide bonds, which are strong covalent bonds between cysteine amino acid in the proteins. So, if there is a protein with a disulfide bond like here, and you add excess beta mercaptoethanol, it will reduce the disulfide bond because it is a reducing agent, and add hydrogen to this sulfur, creating a thiol in the reaction. This process is crucial in denaturing proteins, making them linear for analysis in techniques like SDS page. Beta mercaptoethanol is used to disrupt tertiary and quaternary protein structures, allowing for individual peptide chains to separate based on molecular weight rather than the shape. Disulfide bonds are formed when two cysteine residues, each with a sulf hydryl group, react with each other. These bonds help to stabilize protein structure, including tertiary and quaternary structures. And what is mercaptoethanol's role? Beta mercaptoethanol, also known as 2-mercaptoethanol, is a thiol containing molecule that can reduce disulfide bonds. It does this by forming mixed disulfide bonds with the cysteine residues, effectively breaking the original disulfide linkage. I hope that is all about this question. I hope it is clear to the students. Yes, thank you. Moving on to the 12th question. The only aromatic amino acid with ionizable side chain is dashed. Which is the only aromatic amino acid with ionizable side chain? Option A, tryptophan. Option B, tyrosine. Option C, phenylalanine. Option D, cysteine. Which of these amino acids, aromatic amino acid, has an ionizable side chain? If you remember the structures of these amino acid side chains, then you will be able to figure it out. Let's take a couple of minutes to find out the correct answer, and I will reveal the correct answer in a minute. Okay, this is enough time for this question. The only aromatic amino acid with an ionizable side chain is, and the correct answer to this question is option B, tyrosine. Tyrosine is the only aromatic amino acid with an ionizable side chain. The only aromatic amino acid with an ionizable side chain is tyrosine. It has a phenolic side chain, this is tyrosine, with a phenol molecule, which can lose a proton and become negatively charged at high pH. The other aromatic amino acids, phenylalanine, tryptophan, does not have ionizable side chain. So no OH group here. The OH group of the tyrosine can lose the proton to become O minus at negatively charged at high pH. Tyrosine, an amino acid, can undergo ionization in its side chain, hydroxyl group, as well as its amino and carboxyl groups. The ionization of hydroxyl group in tyrosine side chain occur at a higher pH than the amino and carboxyl group with a pKa value of around 10.9. This ionization makes tyrosine a key player in protein structure and function, contributing to hydrogen bonding and electrostatic interactions. Now, what are the ionizable groups in tyrosine? Alpha amino group, alpha amine, this group ionizes at low pH at around 6. Carboxyl group, alpha carboxyl, this group ionizes at low pH around 2.2. Phenolic hydroxyl group, side chain, this group ionizes at a higher pH around 10.9. This carboxyl is 2.2, amino is 8, and phenol tyrosine is 10.9. So tyrosine is the correct answer, with the OH group forms an ionizable aromatic side chain of an amino acid. I hope it's clear to the students. Okay, moving on to the next question, question number 13. The technique used in identification and quantification of preservatives and sweetening agents in food is dashed. I'll read the question again. The technique used in identification and quantification of preservatives and sweetening agents in food is dash. Option A, X-ray. Option B, SDS page. Option C, ELISA. Option D, TLC. Is it X-ray, SDS page, ELISA, or TLC, which is used for identifying and quantifying preservatives or sweetening agents in food? Okay, I think it's time to reveal the correct answer to this question. The technique used in identification and quantification of preservatives and sweetening agents is TLC, or thin layer chromatography. Thin layer chromatography can be used to identify and analyze preservatives in various products like cosmetics, pharmaceuticals, and food, according to a study on determination of preservatives in cosmetic products and another study on food analysis and quality control. TLC allows for separation and visualization of different preservatives based on their interaction with the stationary and mobile phases on a thin layer of absorbent material, such as silica gel or aluminum oxide. Extraction. Preservatives are often extracted from products using solvents like methanol or chloroform. Separation. The extracted solution is applied to a TLC plate and developed with a suitable solvent system such as methanol, water, ammonia, or ethyl acetate, methanol, or water, according to a study on methyl paraben and propyl paraben determination and another study on loratadine and preservatives determination. So I have mentioned here examples in which TLC has actually been used to quantify real preservatives in products. Visualization. Preservatives are then visualized using UV light or specific detection reagents, allowing for their identification and sometimes quantification. And analysis. The retention factor or the RF value of each spot can be used to identify a specific preservative present in the sample. So this is how thin layer chromatography can be used to detect the presence of preservatives and sweetening agents in the food. Thin layer chromatography looks something like this. This is a TLC plate. Then there is a mobile phase and a solid phase. Sample spots on the TLC of all these purified compounds in a TLC chamber. And then this is the solvent front to the level to which the solvent rises from the mobile phase. And this chromatogram is then viewed under UV light. I hope it is clear to the students that how we use TLCs for detecting preservatives and sweetening agents. Okay, great. Now looking at the 14th question, the histone acetylation catalyzed by histone acetyl transferases reduce the positive charge associated with dashed. So this is a question about epigenetic modifications. Histone acetyl transferases reduce the positive charge associated with dash. Option A, DNA. Option B, phosphate. Option C, histones. Option D, RNA. Which of these gets a reduction in positive charge when acetylated by HATs? This should be an easy answer for you guys. Let's wait for a minute and come back to the correct answer to this question. Okay, enough time for this question. Histone acetylation catalyzed by histone acetyl transferases reduces the positive charge associated with, obviously, option C, histones. These acetyl transferases transfer acetyl group onto histone tails. So understanding quickly how does this acetylation creates a difference in charge. So the histone octamer is positively charged and acetylation actually occurs on lysine residues present in the histone tails. Now when the acetylation brings in a negative charge, it neutralizes the positive charge of a lysine, creating more negative charged histone octamer, leading to repulsion between the negatively charged DNA and ejection of the histone octamer nucleosome. Acetylated histone thereby leads to opening up of the DNA or unwinding of chromatin present around the nucleosome. Histone acetyl transferases are enzymes that catalyze the acetylation of lysine residues on histone proteins, a process that modifies chromatin structure and influences gene expression by disrupting the interaction between DNA and histone tails. What do they do? HATs transfer an acetyl group from acetyl coenzyme A to specific lysine residues on histone proteins, leading to the formation of epsilon N acetyl lysine, like this on the last lysine, nitrogen of the lysine. How do they work? This acetylation neutralizes the positive charge of lysine, which can disrupt the interaction between DNA and histone tails, potentially leading to a more open and accessible chromatin structure. And what is its impact on gene expression? The altered chromatin structure resulting from histone acetylation can influence gene expression, potentially leading to increased or decreased transcription of specific genes. Generally, histone acetylation is associated with increase in gene expression or opening up of chromatin or euchromatinization of the chromatin. I hope it is clear to the students. Very good. Now looking at the last question of this session as well as this whole course live TA session, since this is the last session of the TA classes. The N-linked glycosylation in eukaryotes is inhibited by dashed. This can be a difficult question. The N-linked glycosylation in eukaryotes is inhibited by dashed. Option A, telithromycin. Option B, clindamycin. Option C, tunicamycin. Option D, chloramphenicol. Which of these drugs can block the N-linked glycosylation in eukaryotes? Let's take a minute or so to answer this question, and I'll be back to reveal what is the correct answer. I think that's enough time for this question. The N-linked glycosylation in eukaryotes is inhibited by the drug option C, tunicamycin. Tunicamycin is the drug that does not allow N-linked glycosylation in eukaryotes. Tunicamycin is a nucleoside antibiotic and glycosylation inhibitor produced by certain bacteria. It inhibits the synthesis of N-linked glycoproteins, which are essential for proper folding and function of many proteins. This inhibition leads to endoplasmic reticulum stress and can have various effects including cell cycle arrest and apoptosis. Tunicamycin specifically targets the enzyme responsible for initial step of N-linked glycosylation, which is the transfer of GLC-NAC to dolicol phosphate within the ER. So inside the ER there are enzymes which are responsible for the initial step of N-linked glycosylation, which is transferring the glucnac to dolicol phosphate within the ER. Tunicamycin is an inhibitor of that enzyme which inhibits the synthesis of N-linked glycoproteins, due to which there is ER stress and which can lead to apoptosis. So this is used as a nucleoside antibiotic very commonly. I hope it is clear to the students. If yes, then with that we come to the end of this session. Completed 15 questions for week 12 and we are done with the class. I'll stop presenting my screen and turn on my video chat. Yeah, so with that we have come to the end of this session as well as the end of this whole course live TS session. Today we solved the last lecture series for week 12 and the problem solving session was good. There can be an extra class on Tuesday 3 to 5 p.m. which I'll be taking on the same link. Please do join and we'll be solving miscellaneous questions from all the lecture week series simultaneously. And all the best for your exams and thank you for joining with me and these live sessions. The recordings will be available on YouTube and the slides will be shared to you via Google Drive through the NPTEL authorities. Thank you for joining and I will end this call right now. Thank you.